How our genes interact with the environmental stimuli. Advances in molecular biology have enabled researchers to look at the actual interactions between environmental agents and genes. It is useful to remember that environmental interactions can be specific to an individual's genetic makeup. Not everyone exposed to a toxic chemical develops the same injury. Broadly speaking, gene-environment interactions can be divided into two major types. The first are those that involve actual physical damage in the form of a mutation in a gene, such as the changes that are caused by ionizing radiation and benzene. The second type are changes, which are commonly called epigenetic changes that are caused by external environmental agents that get into the cells and play with the switches that turn various genes on and off and sometimes get stuck in the on position. These external environmental agents are commonly called epigenetic agents. For example, maternal smoking may prove to be an epigenetic agent. There is a known link between childhood obesity and maternal smoking during pregnancy, but the exact mechanism is not yet known. We do know that maternal smoking constricts the blood flow from the mother to the placenta, thus reducing the supply of oxygen and nutrition available to the developing child. It is also known that when the developing child is not getting enough nutrition from a mother, nature can turn on a gene switch or a combination of switches that put the developing baby into caloric overdrive to survive the ordeal. If the switch gets stuck in the on position after birth, it could cause the child to continue to pile up available calories into fat, leading to obesity, hypertension, cardiac problems, and diabetes as the child grows up. By reducing the availability of nutrition to the developing child, maternal smoking very likely triggers nature's survival mechanism leading to obesity, hypertension, cardiac problems, and diabetes in later years. Moreover, there are epigenetic changes that can have devastating effects on the body. For example, phthalates, bisphenol A, and other endocrine-disrupting chemicals can tinker with gene switches that affect the developing baby's reproductive system. Confused hormonal signaling pathways may cause sexual identity problems in childhood, attention deficits, aggressive behaviors, low sperm counts in adult males, infertility, early puberty, and many other problems within the reproductive system. Disease is now suspected to be influenced in part by early environmental exposures include diabetes, cardiovascular disease, dementia, Parkinson's disease, and cancer. The developmental origins of adult disease theory represents an extension of landmark observations made by Professor David Barker and his colleagues at the University of Southampton, United Kingdom, who found that the prenatal nutritional environment can influence health throughout the lifespan. Increasing incidence rates of certain cancers and also of Parkinson's disease and Alzheimer's disease are of great concern. These trends are too rapid to be of genetic origin. The findings raise the possibility that environmental exposures, including exposures in fetal and early postnatal life, may be contributing to non-communicable diseases in adult life, even into extreme old age. A possible mechanism connecting early exposures with late effects is that early exposures may initiate cascades of changes within cells that can lead ultimately to malignancy or that can reduce the numbers of neurons in critical areas of the brain to levels below those needed to maintain function in the face of advancing age. Some of the changes may be mediated by epigenetic modulation of gene expression by toxic chemicals or other environmental exposures. Copyright Academy House www.academy.house